Welcome friends. We are talking about vaccination and we have just discussed about the introductory part about vaccination. Now in this video we'll be taking the examples of these six types of vaccination, and we'll be looking the properties of utilization of this vaccination, a small video. Now here say we'll be looking at three different properties of all these techniques of vaccination actually. Now what are the properties? First one is what kind of immune response they are generating. So the type of immune response that are generated. So this is going to be the first property we need to check. Second property we need to check is whether how much stable it is. Whether it is much more stable or not. The third property we are going to look at is the effect of this vaccination can be reverted or not. Reversion, is it possible? Yes or no? We are going to look at three different properties of these vaccination types. We mainly focus on this C1, live attenuated, then DNA, and also conjugated. We will be focusing on these three parts because this one from this whole bacteria, one from this subunit part, another from is the DNA part, because RDT is more or less the same. We will be focusing on these three major categories. Okay, so now let us first talk about for the live attenuated vaccines. Now the type of immune response generated by the live attenuated vaccines of two different types. It is of cellular mode as well as it is of humoral mode, both. Okay, but in case of conjugated type we are going to see that this type of system where we are having conjugated, the antigen is conjugated with sugar residues and so on, they are going to trigger the immune response only in, sorry, 1 minute, we are going to see, yeah, they are going to trigger the immune response using humoral mediated system. Okay, so because we know that we are having two different types of immune response, one is cellular, another one is humoral. Cellular mode is dealt by macrophages, killer cell, dendritic cell, lymphocytes and all this, sorry, and excuse me, but okay, sorry for that. But in case of humoral response, we are having antibody mediated response as well as complement system. Now in this conjugated form, humoral response is the most important part, but in DNA we also see it relies both cellular as well as humoral. So it triggers both type of response, cellular as well as humoral. Now let us see for the live attenuated form. The amount of stability for the live attenuated virus is very less. So it is very less stable. So it's less stable. In case of conjugated form, it is moderately stable. So we can write it as a stable. But in case of this DNA type, it is very stable. Okay, because we know DNA is very stable and it is directly changing the DNA sequence by combining with it, right? So it is very much stable, the DNA vaccine. Now let us look for the reversion. So whether it is going to revert or not. Now the live attenuated form can revert, so we can put a yes, reversion is possible. In case of conjugated type, the reversal is not possible. In case of DNA, it is also not possible. So these are the properties of this different type of vaccination. Now how they are made. So I have already discussed this. Live attenuated means it's a whole bacteria. Live, killed means it will be the bacteria, but it will be killed using heat or radiation like gamma radiation, because we need to penetrate the cells, right? X-ray cannot penetrate, so gamma will penetrate. Now conjugated type, it is the antigen only will be conjugated with sugar moiety or peptide moiety, then we introduce into the cell. diphtheria. In case of the subunit type of toxin, this type of also is a small antigen, and this antigen will be used to produce antibody against it, and then we'll utilize the antibody solution as a vaccine. Now the DNA type, last one, is again using recombinant DNA technology. We can design the DNA, use the DNA, purify the DNA, and make it a vaccine. A solution of the DNA can be this toxin. Okay, so these are the properties. Now from the next video we'll be talking about details about one of these methods. Okay, thank you.